that did lecture questions 11, 12, or 13. Could you put that into chat? If you covered that on Tuesday with your group and you're willing to talk about the answers to those questions.
I'm sorry, I missed that. Can you say that again?
Is there any trouble hearing me here?
No. I was texting my group to tell them to come join into the Zoom. Okay. My attention was diverted.
That can happen. Okay. I'm just asking if there's a person who's willing to be a spokesperson, because I know I assigned lecture questions. I think I did, at least. Maybe I didn't. I've overlapped these so many times that I think I did. Questions 11, 12, and 13. If not...
I'm happy to help out if you need me.
Okay. All right. Maybe we'll do that. We'll start with you, Dina. And if you want to take on 11, I know 11's kind of a big question. I'm going to, again, take notes and share the screen. And that may get us moving, at least, so we don't fall behind while other people are.
Okay, let me see here.
Let me get into the share. Okay, we should be able to do that here.
I've got too many things open. Hang on. Oh, my goodness. You said it's a big question. I don't have much of an answer for it. That's weird.
Oh, and I'm having trouble. It's telling me it's unable to connect.
Oh, no.
All right. Well, I guess we'll just have to use the awkward annotate function, stop share, on the screen, on Canvas.
I mean, it looks like I just did a real basic sketch of stimulation of a B cell.
All right, hold on. Let me see if I can get into the annotate part. I think I have to share screen again and do a whiteboard. And then I'll just try and take notes, but I can't write very well with this thing, so I'm going to try and just type what you're saying. Okay, so the question is asking you to compare. So what are some similarities between, sorry, I can't type, B versus T cell in general? Doesn't matter which T cell.
Okay, well, in general, the dendritic cell must engulf, process, and display an antigen via its MHC2 on its surface. That would be for the CD4, and I believe for the B cell, and for the just general purpose on the MHC1 as well for the CD4 or CD8. That displays it to the CD4 cells, which trigger the activation sequence. The activation sequence results in the helper T cells needed for the B and CD8 cell activation and co-stimulation. And I think I just sketched over it because I went into so much depth on the CD4, CD8 activation.
That's the next question, yeah.
Yeah.
Right. Yeah.
Well, I mean, earlier in the discussion on like questions 6 and 7, there's questions 7 and 8, I went into a lot of detail on.
All right. Well, let's expand a bit on this. So the dendritic and macrophage, there has to be an antigen presenting cell involved. And the last time we did talk about potentially 4 antigen presenting cells. In the case of the CD8 T cell, it can, although the book doesn't really talk about this, it can be initiated with a body cell presenting this, but it won't co-activate. I have this, I don't know what idiot put this cord right where I sit, so I'm going to have to move because I keep getting tangled up in the cord. So they've been in my office doing stuff since I was gone, so I don't know why they did that. But at any rate, so you need to have an antigen presenting cell, and dendritic and macrophages are common antigen presenting cells that are involved in both these activation. But in the case of the B cell, so let me maybe, if I can make a new text box, I think I have to go out It's been a while since I've done this, and go back in and put the text box somewhere else. So differences. The B cell acts as an antigen presenting cell, and we'll go through how that works. But that doesn't mean that the dendritic cell is not involved, because in order to get the B cell activated, it needs a T helper, almost always. There are a few exceptions. And to get the T helper, you had to have had a dendritic macrophage be an antigen. So I guess I get new ones all the time. All right, so let me move it over here. So what other similarities? Well, we have the, I'm going to move this guy down a bit as it looks like it'll right over the top. It also is telling you that the MHC has to be involved, in that both B and T cells require their particular CD surface molecule to bind to the matching, the one that they match with of the MHC. So last time we said there are CD4s and they bind to MHC2, and there are CD8s and they will only bind to MHC1. The other part, which I think Dina skipped, was you have to have the appropriate antigen receptor bind its antigen. So this process, and let me see if I can, these processes here are what we're going to call double recognition. So you can see this, two things have to happen. Whether you're a B or a T cell, you have to have your MHC bind, and you have to have the appropriate antigen bind the antigen receptor. That's what they mean by double. There has to be two binding events, and those are happening because the MHC is holding on to the antigen. And then the CD4 or CD8 molecule is associated or stuck together with the appropriate receptor. So they come into close contact, and they're able to simultaneously send their CD8 over to bind the right MHC and their antigen receptor to bind the antigen. That is double recognition. So to get almost all, and this is where, there's a few exceptions, but more than 95% of B cells require this. A few B cells don't require double recognition, but only require an antigen to bind its antigen receptor. But that is a rare few antigens that work that way. So overall, you need double recognition. And I may have missed anything else that Dina said, but the other thing that is pretty much required, but the definition of which, or exactly how it happens, varies, is you need something called co-stimulation. So what that means often is, but not always, it means some sort of exchange of either interleukins or co-stimulatory binding proteins between the antigen presenting cell and the T or B cell being activated. So I'm going to just say often via interleukins or co-stimulatory protein on the antigen presenting cell to the lymphocyte, I guess, lymphocyte being activated. So the reason I'm having this question 11 is because it's setting sort of the ground rules up for the activation of nearly all of the lymphocytes of the adaptive immune response. Now, the one exception to that when we get to B cells is there's a very few B cells that will just bind their antigen and undergo clonal selection. But even those guys, if interleukins are supplied by T helper cells, will be a more vigorous response. Now, the other part here is what happens, and I think that's later. Let me see. Yep, that's later. So we'll worry about that, the other part later. Well, what about these differences? So 4 differences. We know the B cell acts as the antigen presenting cell. So that's versus the dendritic or macrophage acting as the antigen presenting cell. And I'm just going to put in parentheses with the CD8 body cells could be, and I'll put it in parentheses because typically books don't call them antigen presenting cells. Maybe instead of saying that, could be the double recognition event. Maybe that's the better way of saying it. That way, we're not all messy with the word antigen presenting cell. So what other differences could occur here? Well, one thing we're going to do soon is we're not going to identify who the receptors are for the antigens on the T cells. We'll just call them T cell receptors. But for the B cells, you will know, you're going to need to know that IgD can be an antigen receptor. And another aspect of the B cell is B involves endocytosis. We've talked about this already, versus T involves, is this not visible to you guys now? Is it still visible?
It's starting to cut off, yeah.
Okay, so let me make this bigger. That may be what the problem is, and I have to move you guys to do that. There we go. Whoop. It's very sensitive, this. Okay, there we go. It involves phagocytosis by the antigen presenting cell, meaning the dendritic or macrophage. Now, clearly there are going to be other differences, what the cells are that they mature into, but one of the things that is common to both is once there's co-stimulation involved, co-stimulation required, although it's different in the two, but CD4 and most B need interleukins. CD8 can use co-stimulatory proteins. I know I'm running out here, so I'm going to write up over here. I hope it'll, yep, it will. Okay. And then, what was the other one I was thinking of? Oh, so the results of both would be is to cause clonal selection, and that certain without knowing the numbers, interleukins increase the rate of mitosis. Finally, and this is something I think question 14 is about, if co-stimulation does not occur, that leads to anergy. I think I spelled that right. So we'll talk about anergy shortly. Let's see how we're doing. Oh, we got 24 people here. Great. All right. So far we're still on 11, and you can see there's quite a bit of information here, but exactly the steps involved are going to be different for the two T cells, CD8 versus CD4, and for the B cell. So the details of how these occur are going to be a little different. Now, what I find in the book is that they go into pretty good detail explanation for your CD4 T cell activation, but they're confusing and sort of for a good reason, and incomplete for the CD8 activation. And then in words, in the text, they explain the B cell activation, but they have no good pictures of that. So be aware of that. And this is why I really wanted to make sure we spent some time today on these questions, because without me going through them, you won't have all the steps for the three cells that we're going to activate: the B cell, the two T cells, CD4 and CD8. So question 12 is comparing and contrasting the activation and co-stimulation of CD4 and CD8 cells. So Dina is started talking about this, but was there anyone who covered this in a group that, let me open chat again, that's willing to be a spokesperson? Or let me see what we've got as a question. IgM can also be in. Yes, that's true. I put IgD without IgM. Yeah. So there are both of those antibodies, but the only role you'll have for IgD when we cover the antibodies is as a receptor that we know of. Okay, so is anyone who went through this in their groups? Okay, so what I'd like to do is, maybe what I'll do is I'll get through till question 14, since we started that, just going along with whoever wants to volunteer. And then I'll make the groups, which will be 15, 16, which are labeled Group C and Group D and Group E. And hopefully that will get us through the rest of these lecture questions before we have to leave. I'm just doing it myself in order to speed things up a little bit. And I know I didn't sort you guys out into those other groups, the B, C, and D ones. So let's see, what do I do with this? I guess I have to, if I stop sharing and start sharing again, will that erase it all? No. Well, dang, this is why I don't like using this. Does anyone have any questions on this before I do erase it? Okay. I know I can erase it by box, but I made many, many boxes. Okay. All right. So if, up to you, Dina, or anyone else, if you want to talk about what is required for the CD4 to get activated. I know Dina already mentioned that you have to have an antigen presenting cell and all the steps that it goes through, making a phagosome and fusing with a lysosome, and then becoming the antigen becoming bound to the MHC2. So I'm going to just say with, and it already has all that happened, MHC2, and I'll just make an asterisk for the antigen. There's the particular antigen bound to it. Now, what does this cell do? Okay, and we'll say this cell is at site of pathogen entry. So then what does this cell do? Anyone want to volunteer? Where's it going to go? I'll open the chat in case you'd rather just, or maybe you can only type it into chat. Is it going to likely do it in the connective tissues or the epithelial tissues that the dendritic fixed macrophage or the the free macrophage engulfed it? Where does it need to go? I know we talked about this.
It's going to go to the, I want to say the thymus, but I'm feeling like
Those T cells already got through the thymus. That now they're
Right,
waiting. Where do they wait?
it's going to, gosh. I mean, the dendritic cell engulfs it, it presents it, and then it matures it. It activates it in the, I know this.
Is she struggling?
Does it
Anyone
not
want to
just
save her?
does it not just go to lymphatic tissue or organs?
Yes.
Yeah.
So good. Thank you, Noah. It migrates to lymphatic tissue. So those slides that you're drawing, if it's picked up into from the interstitial fluid into the lymph, it'll end up in a lymph node. If it manages to get into the blood, maybe because capillaries and arterioles are cut near the site of infection, it might get done in the spleen. If it is in a mucosal membrane, then those the lymphoid follicles, like the Peyer's patches, there you're going to have T cells, which will include the CD4 cells, residing and waiting to find their antigen presenting cell with their antigen on it. So it migrates to the lymphatic tissue and finds the CD4 cell with a matching, so I can't type, can I type and talk, receptor for that antigen. I'll just say that guy. Okay. All right. Now we undergo what the processes I brought up before, right? So tell me what happens in co-stimulation, in double recognition. Who binds to whom?
The APC is basically just going to present its antigen to the appropriate specific CD4 antigen receptor, and they'll bind together, and that causes double stimulation.
That causes double recognition?
Right, double recognition, yeah.
Part. Oh, you want co-stimulation. Okay, but you're jumping over what's actually binding. So the MHC 1 or 2 is involved.
It's going to be 2 since we're talking about CD4 cells. Yeah.
Therefore, that binds to the CD4.
Right, and that would be the double recognition, right? With the MHC and then the antigen.
Plus antigen receptor on the CD4. Binds to the antigen on the MHC2. So you have this complex. There's a nice picture of this in the book. You can see it happening as they come together. Now that means that why use the lymphatic tissue? Well, that's where lots of T cells of a lot of variety of receptors are found. If it doesn't find it in that one, it may keep wandering to another lymphatic tissue until it can find its matching CD4 cell. And this has to happen before almost any part of your adaptive response can occur. That's one reason the adaptive is slow to initiate with a novel pathogen, with one your body's never seen, because you might only have one CD4 cell with that particular antigen's receptor on it. Now you might say, well, that's worrisome. I can get sick all the time. Well, luckily, most pathogens have multiple antigens on them. So you don't have one dendritic cell becoming an antigen presenting cell with one antigen, finding one CD4 cell. You have a variety of antigens that are being introduced. So if we have time, which I know it's a bit rushed, but that is a good discussion around vaccination strategies and why vaccination may not be as long lasting as actually getting sick with the disease. Because when you get sick with the disease, you activate a whole array of CD4, CD8 potentially, and B cells. So once you are ill, you have a large variety of memory cells being generated. If they're using these very isolated antigens and making a cocktail like the flu vaccine I got yesterday, they may not have all the antigens that would be on the actual pathogen. They're using key ones that they think will strongly stimulate your immune response. So it's something that can wear off because you're not stimulating and generating that many memory cells. There are other strategies where you can use dead or killed or even just weakened pathogens. Or sometimes, like tuberculosis, they use a very close relative that doesn't get you sick. And they use that to try and have more of an array of antigens being exposed during the vaccination process. So, but you can see here that if you just have this one antigen presenting cell, it may take, well, as you'll see with the antibodies, it might take 10 days or 2 weeks before you actually can activate the full adaptive response. Okay, so once they do double recognition, as Noah said, you need to have co-stimulation. And in this case, what happens in co-stimulation, and you don't have to worry about the numbers on these things, but if you want to, you can tell me. Who gives what?
The CD4 will give the APC interleukin 1, I believe, and then the APC will give interleukin 2 to.
Right. Okay, so we have an antigen presenting cell releases, and I don't care about the numbers, one interleukin, and then the the CD4 releases a different interleukin. So they exchange interleukins. So the part that's a bit hidden and not really discussed about this is that's the most clear idea of co-stimulation. I mean, you can imagine, it's like somebody stimulating you with a picture, and then they stimulate you back with a different picture. You're both getting excited by looking at these pictures. So the same idea, they're giving each other, interleukin cytokines in order to change each of them. So it's clear how the CD4 changes. What does the CD4 start to do? It starts to do mitosis, right? And what is that called?
Goes into clonal selection.
Clonal selection. And eventually it's going to make we'll only talk about 3 general types. So that's the T what?
Helper cell.
Helper. And
Regulator.
T regulatory.
Cytotoxic.
More CD4 memory. I'll put multiple here because that'll help you remember why it might be faster the second time. Now, what is hidden, so I'm just going to go through it. I know I did it in the lecture, but is that the dendritic cell is changed as well. If it's a macrophage, I don't think this happens. But if the antigen presenting cell is a dendritic cell, it begins to express what are called co-stimulatory proteins. So it's now experienced as well, expresses co-stimulatory proteins on its membrane. And the one that is, I think it's mentioned in the book, but I know I lecture on it. For example, B7. So I do want you to know B7 because we're going to use this in a moment for the CD8 co-stimulation. Okay, so you get many T helper cells, you get Treg cells, and you get memory cells from this guy. So that is how you start all this process. I think a later question, which I'm probably going to assign, talks about what do all these cells do? And we kind of covered that already, but we can come back on it again. So this was the CD4 cell. Can I change color or anything with this thing? Format, I probably can. Yeah. Okay, so let's do the CD8 in red here. So this is where things are not well understood or not well described in the book. And the picture that they show is sort of a combination of more than one scenario. So let's do the scenario where you use a T helper cell, and you're going to use a dendritic T cell, or an antigen presenting cell, antigen presenting cell, and we'll just say it's a dendritic. This is what matches what's in the book, the picture in the book. Activates CD4, and it becomes a T helper. So all this happens first. I'll put 1 T helper. We have to have that happen. It has to have a T helper available around because we're going to use the interleukins from it to allow co-stimulation to occur for that CD8 cell. So now this is where the picture in the book shows 3 cells coming together. One of them is a dendritic and the T helper cell are together, and the other is the CD8 T cell. And it shows it binding to whom? Binds its MHC1, binds its T cell receptor and CD8. Here we go. I'm going to have to move this, I guess. Is there a way for me to move it? Yes, I'll put it up here. So it has to do, it's required that it does double recognition. So the CD8 has the, let me get rid of this guy over here. The CD8 cell has its CD8 T cell receptor for this guy, right? We'll use the same thing for the antigen. And it has to bind its CD8 to what kind of CD8 molecule binds to what? This is our double recognition. MHC1 or 2?
MHC1.
1.
All right, so this is where that statement that dendritic cells can be unique in that they can take an exogenous antigen and they can put it on both MHC1 and MHC2. So here they have that antigen was exogenously attained, it was phagocytized. But that dendritic cell did not only put it on their MHC2 in order to undergo this process, but it also put it on its MHC1. So I'm going to just put that in parentheses. This can happen if antigen presenting cell is a dendritic cell and it has put this antigen, I'll put exogenous, onto both MHC2 to activate the CD4 cell and onto MHC1 to activate the CD8 cell. So nowhere in the book does it actually say that. It just shows this picture where that is happening. Are we okay? Any questions?
A quick question.
Mm-hmm.
It can still go up because the T cell, the CD8 cell is wandering, right?
I mean, right now it's wandering when these.
Yeah, is it wandering or is it once it's active that it wanders? Because it can bind to any MHC1 that has its appropriate antigen, right?
Right.
Okay, it doesn't have it's just the dendritic cell is a special case where it can do both.
Yes.
Okay.
So you're bringing up or alluding to some key part of this, and that again is not talked about in the book. CD8 cells have multiple ways to become activated. All those ways require double recognition and co-stimulation. So I'm not going to test you on this, but just to give you an idea of why that would be needed. Let's say you have an infected body cell with a virus. A CD8 T cell that matches that antigen encounters it out in your mucosa because it's wandering around, or there's an inflammation event, because like your throat gets sore when it's infected with a virus, that's inflammation. So that CD8 is called out of the blood. It was in the blood. And it finds its antigen presenting cell and undergoes double recognition out there with the body cell. The body cell is sort of acting like an antigen presenting cell, but it can't do anything yet because it needs co-stimulation. So now it can wander back into the lymphatic tissues, and what it needs to find is it needs to find a co-stimulatory interleukin cytokine, which could be provided by a activated T helper cell, or, and this is what the book also shows without totally explaining, it can find the dendritic cell, and that dendritic cell that was interacting with the CD4 and expressed a B7 over here, that B7 can cause that CD8 cell to become activated. So I'm going to put this as alternatively. If the CD8 is undergoes double recognition, out in the tissues, or maybe out in the tissue by an infected body cell, it still requires co-stimulation. To get that, it can either go into the lymphatic tissue and either be co-stimulated by the appropriate, meaning the same type of appropriate T helper, that is the one for that particular antigen, or by a dendritic cell that has interacted with a CD4 and now has B7 or other co-stimulatory proteins on its surface. So there's no B7 until that dendritic cell has encountered the CD4. They did their co-stimulating. They changed, the dendritic cell changed, and therefore it now has B7. So that allows the CD8 to get co-stimulated by this B7 or other co-stimulatory proteins on the surface of the dendritic cell. And you actually don't even have to have a T helper cell and interleukins. However, if you have both, yikes, it's running out of room again. Let me drag it up and make it bigger. However, if both interleukins from T helper and B7 occur, you get a more vigorous response, more vigorous clonal selection. So you're going to get more, yielding more T cytotoxic and T memory. So one way I like to think about this is that the CD8 cells are going to be making soldiers, right, that go out ultimately to kill infected body cells or cancerous body cells. And therefore, there are a lot of various ways in which you could generate these, because there are different situations in which that particular antigen can be presented. And so it has options. And I'm only very crudely going through these options because, as it turns out, there's lots of types of CD8 cells and other, things that you don't want to know about, I'm sure. So we have the answer to 12. These are the toughest, probably, lecture questions that are in this whole set. So I want to stop here and make sure there are no questions about this. Let me get the chat. Yeah, there it is. Around before we go on to 13, because we've actually answered 13. Here is 13. All right, so you're either totally mystified by this whole thing, and I wish I could say, even though generally I think the book is pretty good on all this, I think this is where it's, because they can't cover it all, it ends up kind of covering little pieces of it without really explaining them. It shows 3 cells coming together. It shows B7 or co-stimulatory proteins on the dendritic cell, but it never really explains it very well. Okay, no questions? All right, let me erase this then. I didn't want to do that until you guys clearly didn't have questions. Okay, so, and I keep moving this thing down because I got so many things on my screen I can't type at the top. So the next one's a pretty easy short answer, because what happens if co-stimulation does not occur?
Nothing happens.
Yeah, and there is a name for that.
I forgot it. I'll be honest, I forgot it.
Energy.
This prefix.
Energy, yes.
Without. Yeah, so I don't know where the rest of it comes from, but it's energy occurs, meaning that B or T cell dies. That's not good, right? You don't want that to happen in a sense, because you might only have one B or T cell that is with that antigen receptor. Why would we evolve this way? Well, because especially with the CD8, it's more obvious, but even with the B cells and antibodies, and even with the interleukins from T helper cells, if you are mounting a huge mitosis event and you're causing an increase in your innate, so inflammation is going on and all kinds of bone marrow transforming into making white blood cells from red blood cells, say, you're going to undergo this huge change in what you're doing as your metabolism in your body. You're going to invest a lot of energy. You want to make sure that you actually have enough pathogen in you to mount an adaptive immune response. So think of a scenario where, one body cell gets infected, say, by a virus, and you are lucky or unlucky, depending on how you think about it, and a CD8 cell encounters that infected body cell, and it becomes undergoes double recognition. So it's MHC1 binds the CD8 and the antigen receptor on the CD8 cell binds the antigen. Everything's great. Now it runs into the lymphatic tissue and it finds no dendritic cells that have been activated with a B7 and no helper T cells to that antigen that's been activated. So it dies. And the reason it finds no CD4 or dendritic cell is probably because you have so few pathogens in you that you haven't activated enough dendritic cells or enough CD4 into T helper. And you might say, well, I don't want to lose that particular CD8 cell, but if your infection level is that low, then your innate response will probably take care of it. Is this kind of making sense? You don't want to put out all this energy unless you know you have enough of an infection occurring in the body to warrant spending all that energy. Are we okay? Okay, so energy means their actual elimination of T cells, that they do die, or B cells, that B or T cell dies. I think the book alludes more to T, but that's okay. All right, so the next thing is to compare and contrast. So I'm going to get rid of this one. And just because we'll run out of time, I think rather than break you up and give you 15 minutes, I think we'll just try and get you guys to give me some answers to these. So cytotoxic T cytotoxic. And then over here, we'll make another box. Let me grab this box if it'll let me. Can I move this box over? Oh, well. I'll delete that box. Let me move that box over. So we have some row T cytotoxic, and then I'll make another box over here. And we'll talk about natural killer cells. Is that what it is? Yeah, natural, NK cells. So tell me about what these cytotoxic T, well, first let's start with natural killer because I got blue. So what do they look for to know who to kill?
They look for a lack of MHC1.
And they don't care about the antigen. They just lack of MHC1. So triggers, or they could have antibodies on them too, which we'll come back to later. Triggers recognition of infected cell. Okay. How about the T cytotoxic? What do they need?
They look for the MHC1 with its specific antigen.
Yeah, so I'm going to just call that, to shorthand it, double recognition with its antigen. So it has to be that specific antigen binding its receptor. Okay, so this one is highly specific to a particular antigen. This one doesn't care. You could have this NK cell could kill a cell with MHC1 and a certain antigen, and its same natural killer cell could attack a different body cell with a different antigen on its MHC1 because it doesn't care about the antigen. It only cares about the amount of MHC1. On the other hand, this guy over here, it could have a diminished MHC1 number. In other words, the cell could be infected by a virus, not making many MHC1s, but the ones it makes has an antigen on it that's a red flag for this cytotoxic T cell. So this kind of covers a range of situations, this one being fairly nonspecific, but also insensitive to specific antigens. This one, if you have many, you could have many MHCs with that particular antigen on its receptor, or you could have few MHCs. It's still going to respond the same way. So that's the main difference between these two, because we're going to see how they respond, which maybe I'll do, since it's common, in another color. So what, whoops, I didn't mean to do that. I guess it got to make a new box first. Okay, so outcome of their responding is going to be what? What do both of them do?
They induce apoptosis through preferent and granzymes.
And they also release some other things.
They also release gamma interferons. I don't know if that's what you're looking for.
They release interleukins.
Okay.
And to activate different interleukins than we had before, but don't worry about that. To activate macrophages and other innate. And you're right, there is an interferon. I think it's only from the cytotoxic T, though. So maybe I should make that a new box. And maybe we'll put it actually down here somewhere so I can see it. So T cytotoxic releases, gamma interferon. And why would they do that? What are their effects? Anyone? What do they increase?
It increases MHC1 expression.
Of it's actually both MHC1 and 2. So if you are a cytotoxic T cell, you release this gamma interferon. I'd have to double check on the table whether you can look at for me. I guess I have the table right here. I think the gamma interferons. Oh, it does say natural killer cell. So both of these. So this should be, I guess, green. Instead, yeah, sorry about that. So NK and these guys release gamma interferon, and that causes this expression to go up. You want the MHC1 to go up because you want any healthy cell to signal it is healthy, and if that cell is not healthy and it's making the antigen, you want it to show its antigen. You're saying basically, show me your normal or abnormal antigen. If it's showing many more normal, the natural killer cell will slow down. If it's showing more of the abnormal, you'll get more effective killing by the cytotoxic T cell. Now, why you want this to go up is because you then can activate more T helper cells, potentially, if there's more antigens on there. You can get these guys, the more dendritic cells sprouting more exogenous antigens and getting more T helper or more B cells. So these are ways of sort of amplifying both the recognition of who should be a target and who shouldn't be with the MHC1, and you're amplifying with MHC2 the other aspects of your adaptive response. Okay, so I want to get through, we have enough time, the B cell activation, and then we can kind of quickly look at antibodies, hopefully. I think that part in the book and what I covered in the lecture slides on antibodies go pretty parallel. There's not much new stuff in there. Or context that I'm giving you. The book does a pretty good job on that part. So let me just make sure we get through B cells being activated, because the book doesn't do such a great job. It has a picture, but it doesn't do such a great job on that part. So this is 16, question 16. So let's go through this stepwise. Stepwise. B cells activation, and just make steps like we did before. And this is going to be vast majority of them. This is where there are a few exceptions. So who do you have to have around if you're going to get, well, let's start with what happens with the B cell. Let's say the B cell, it's going to bind the antigen to what? We already covered that, so that's maybe where we'll start. And this is the one that Noah, you corrected me in the chat. It's receptors.
Oh yeah, it's going to use IgD and IgM to bind to its an MHC2 APC.
And or.
IgD, right? IgD and
IgD, you're right. Typo. IgM, important typo. So now for IgD, when we get there, this is the only function we're going to have. It is secreted in tiny amounts, but it's not known why. This guy gets secreted and does other things, but apparently both could act as a receptor. So remember, these are antibodies, and so antibodies are highly specific to a particular antigen. So each B cell has different type of IgD or IgM receptor, and it's got to bind it. And then that leads to the B cell undergoing endocytosis, which we've talked about already, and eventually binding the antigen, maybe I'll just do that, to MHC 1 or 2. Anyone again? Is it 1 or 2?
Could be 2.
2. i.e., it becomes an antigen presenting cell. All right, we got to present this antigen to somebody. So in order to present this antigen to somebody, we're in fact going to present it to a T helper cell. We had to have gone through all the steps for the dendritic cell to find a CD4 cell to cause clonal selection. So I'm going to say all steps to convert that same CD4 to that same antigen into a T helper. So the steps on the previous slide that we did of the CD4 and how the dendritic or macrophage presents its antigen, there's double recognition occurring, and that double recognition leads to co-stimulation, interleukins are exchanged, and now you have a T helper cell, and that T helper cell has to match. It has to be for that antigen. Because what are we going to do next, which we always are required to do? When these guys find each other, what do? Starts with the word double. Big hint.
Double recognition.
Double recognition. That is the CD4 protein on the T helper binds to the MHC2 and the antigen on the MHC2 on the B cell binds to the antigen receptor on the T helper cell. Okay, we have our double recognition, but if we don't get the next thing, anergy is going to result. So the next thing is automatically kind of going to happen here, but what's needed now?
Co-stimulation.
Co-stimulation. And that's going to be via interleukins released, and don't worry about the number, by the T helper. So you can see why energy doesn't happen as much with the B cells, because their T helper cell that's going to co-stimulate them is bound to them. And it's not there's not as many situations where you would have double recognition occurring without there being either a T cell buy or a dendritic cell buy to do the co-stimulation, as there are in the CD8, where you can get double recognition by an infected body cell and then never find your particular dendritic cell or T helper cell. Here they're right there, so it's all good. That leads to clonal selection. And what do you call these cells that are made?
Plasma cells and memory B cells.
And the plasma cells release antibodies and memory B cells. Okay, once again, multiple memory B cells are now made to that antigen. So the next time around, our secondary response can be faster. Okay, that is the toughest part too. And it looks like we still have about a half an hour, if I'm right, to finish up the last questions. So,
Professor,
yes.
I have a question.
Mhm.
Can you clarify the difference between the step 3 and the step 4? The step 3 also goes for the double recognition, right?
Yeah, you're right. So in step
3 and 4. No, 3 and 4.
3 and 4. Yes, but hold on. In step 2, what's happened is you became an antigen presenting cell. So in order to present your antigen to somebody, you need that to be presented to this T helper cell down here, right? And where you're binding this.
Right.
Yes. Well, in order to get that T helper cell, you had to have all the steps to convert the CD4 cell. So I erased those because I'm not using my tablet since it doesn't work here.
Yeah, I have that copy.
That's on the previous page. So that's where your dendritic cell or macrophage does double recognition, co-stimulation, and you get. So that's what step 3 is referring to. All the steps on the previous page.
So step 3 refers to become the T helper for double recognition.
Yeah, so no matter what, if you are a lymphocyte, you're a CD4 one, a CD8 one, or a B cell. With the exception of a few B cells, you require double recognition and co-stimulation in order to undergo clonal selection.
True. Okay, thank you.
All right, then I have another question. Did we go over question 13? We did when I had the 2 columns. 13 was about how you activate a CD8 cell. And early on, I had a column where we activated the CD4 and then all the steps to activate the CD8. Yeah, so I did go over that. And I'm sorry, because the tablet doesn't work, I can't scroll back and forth and show it to you again. Okay, so let's, I'm kind of thinking we have a half hour. How many questions do we have? We have 2 groups. We'll use half of it. Well, let's just keep going as we are since I can't take notes easily. And if I give you 15 minutes to do these last 6 questions, I'm afraid we may not finish today. So let's go on to the 17, which is the effects of antibodies. And again, ideally someone, I know Noah and Dina have been the real workhorses here, but ideally, what can antibodies do? So 6 things. Maybe other people shout out some things. You know about some of them because we just did a test in lab on one of them. Maybe I'll just pick some names and pick on people. Okay. Maybe Eric, can you tell me one thing they do out of the 6? I don't care which one. Maybe he's not there. How about June? Can you tell me one?
Agglutination.
Agglutination. Now, there is only one antibody that does it. It's IgM only. Can do that one. Someone else, who else?
There is answers in the chat.
Ah, okay. I can't see chat right now. Thank you, guys. Okay, so we got neutralization, and a couple of them can do this, IgG and IgA. And then opsonization. Oops. Okay, I have to capitalize. Once again, IgG and IgM. And what else have we got? Plano is what, we'll put that over here in another color or something, another text box over here. There's an abbreviation that is in the lecture slides. So far we've got opsonization, neutralization, agglutination. What does P stand for? Precipitation.
Yeah.
And that is IgG only. And sorry, I messed up with my 5. And L. It's the same thing L stood for in complement.
It's going to be lysis.
Yeah, but and it is actually going to be via complement activation. And what pathway? Classical, alternative, or lectin? Classical, yay. So we're really talking about the MAC attack, and that MAC attack is triggered by complement, and complement can be activated by IgG or IgM. Okay, so we have precipitation, lysis, agglutination. What do you suppose I stands for? Something in the innate. We got it, inflammation. And that can happen several ways. It can happen via complement, C3A activation, and that would be IgG or IgM, and or via IgE. And IgE is an interesting antibody, as we'll soon learn, because it, like C3A, it also binds to basophils and mast cells and leads to more histamine. So there we go with this way of trying to remember what all these stand for and all these things it can do. Now, we didn't include this because it's not in that figure, so I'm not going to number them. But you also heard that they can help label cells for destruction. And that's with things like the natural killer cell, for example, or via, did we do opsonization? We did do that, opsonization. I guess that's already opsonization, which would lead to increased or even the ability to do phagocytosis. So these are powerful tools. And the one thing nice, as you can see, is that really, other than inflammation itself could be a little damaging, most of these things are not damaging to your own body, but excessive inflammation can actually damage you a bit. But let's make sure we know what all these things are. So over here, I'm going to move my chat window over here, so hopefully I'll still see that. Let's Let's do another text box, and we'll do it in another color again. Let's talk about what these things are. Agglutination. So this is a sort of form of clumping like you have with precipitation, where the antibody bridges multiple antigens into a group, but in this case, it's cell-bound antigens. Into a group. And where did you see that in the lab for the first lab test, for example?
Blood typing.
Blood typing. So blood typing used the IgM. IgM, and I would know that there are 5 subunits and therefore 10 binding sites. IgM has 10 antigen binding sites. And it does that by having those Y-shaped molecules. There's many of them all bound together. Okay, so that's agglutination. Close relative to that would be precipitation. But now you're doing acellular, or soluble. Sometimes they call them soluble antigens. So much smaller antigen, and it complexes together, and you saw that in the lab as well. So neutralization is a broad term, and that may mean it blocks binding of the pathogen. It could bind or binds toxins and detoxifies. So you could think of that being something like the pathogen's trying to eat you, it releases enzymes, the antibody binds the active site of the enzyme, no longer an active site. That's neutralization. So it's kind of an umbrella term for lots of things that prevent the pathogen from doing the destructive processes that it might do. So if it's a virus, maybe it binds the virus in a way that the virus can't enter your body cell. Okay, I think the rest are explained. If there are questions about that, let me know before we go on to the next. So one of the things I would like you to do, there's a nice figure in the book showing you, all this inflammation, all this the antibodies can do, but it doesn't list which antibody does which. And you should know all the actions of each of these antibodies. So we're going to come to a little bit more in a minute, but I did that for you kind of here, right? So you should know that inflammation can be done by IgE or indirectly by IgG or IgM through complement. So if I asked, on a multiple choice question, which of the following antibodies can do neutralization, you'd be able to tell me which ones can do neutralization. Okay, I don't see anything in chat. Let me erase this so we won't run out of room. Any questions before I erase it?
Real quick, Professor, which of the antibodies label cells for destruction? Is it the same ones that do opsonization?
Probably IgG, but I don't know if IgM does. So you're right, I didn't name that one. But IgG is the one that's going to circulate, and IgA is, did I say M? IgA is in tears and body secretions. So I'm not sure that it has that, but IgG I know does.
Okay, thank you.
Okay. All right, so there are other skills that these have. I just put this one down, but let's talk about those in a minute. Do I have the eraser? Yeah. Okay. Okay. All right, so text. All right, so if we go on, the 6 possible effects, we did that. Name 3 binding sites. So the one we're going to be talking about is on IgG. because it has all three. Not all of these have all three types of binding sites. I don't mean that there's two antigens. These are different types of binding sites. So one is, I'm going to give away, it has at least two sites to bind its antigen. Because it's that Y-shaped molecule and at the end of each Y. But if you're in IgM, you've got 5 of these bound together, so you've got 10. And you'll see some of them have 4 and some have 2. It depends on which antibody. But there are other sites, and one of them is you have a macrophage binding site. And that would be sites on IgG and IgM. And what does that allow you to do from the plano?
It allows for opsonization.
And similarly, if you're the same two, you have a complement binding site. And that allows classical activation. So if you are an IgA, you're not going to have all of these other ones. Now, where these sites are is if my hand is the Y, there's a Y, I guess. Here's an antigen on that stalk of the Y, you can have these other binding sites. Okay, are we good on that one? All right. Okay, so it says for each of the 5 types of antibodies, give all their functions. Well, we've kind of done that, but if you want to see it, maybe let's do this and add something to it.
So I have a quick question.
Sure.
In your lecture notes, you put that the macrophage binding site is only on IgG and not IgM.
Oh, you're right. You're right. Sorry, I couldn't erase just that. It erases the whole thing. Yes, I'm now seeing that in my notes too. So misremembered that. So actually, I made a little tiny. Maybe I'll write that. Oops, I need to go back to this. I had IgM does complement and agglutination. I don't know if this will help you guys. I just made this little tiny note for myself. Does neutralization and precipitation and IgG. Oops, G. So I may have been rushing to try and get us through everything. Opsonization. And we'll come to this later, passive immunity. And this guy does passive immunity too. So IgE does inflammation. So that's complement. That's what the C stands for. Neutralization, precipitation, and opsonization. So just to make sure, these are my little notes to myself. Okay, so there is one other antibody here, IgD, right? And that's the receptor on the B cell. So because I put that here, IgM would also be.
I also have another question.
Yeah.
I thought by passive immunity, do you just mean it circulates in the blood?
No, we'll come back to that. So hold on to that question.
Okay.
I'm just giving you kind of a little crib sheet, not that you should use it in the test, but to help you remember which ones do which. Okay. All right. So, but remember, you got to translate this crib sheet. This inflammation is from mast cells and basophils causing greater inflammation. And this complement can cause inflammation, and it can cause opsonization, right? So complement activation can indirectly cause inflammation and opsonization. Okay, I don't know if that'll help you, but at least I put that over in my margin to help me quickly remember, which clearly I should have checked before speaking earlier. Now, one last thing, and I put these in this order for a reason. This one is always first to be released. So what happens is when a B cell becomes a plasma cell, it's not like you have one plasma cell releasing this and other plasmas. They all do them, and they do it in a certain order. So any plasma cell I picked out would first secrete, maybe that should be an ST instead of first degree. First, it would secrete IgM. It wouldn't be making IgA or IgG at all. At that point, it would have IgD on it because that's its receptor, but the others wouldn't be being made at all. Then these two are released second, and not in equal amounts. So we'll take a look at amounts, but 70%, maybe I'll type that, 70% of what is secreted is going to be IgG. So you're only making a little bit IgA and a little bit of IgM. Now the IgA ends up being secreted into the blood, but then picked up by the epithelial cells of glands and ends up in body secretions. So it's going to be in the mucus. It's going to be in, maybe I should have typed this because I can't write very well, secretions, in tears and saliva. That's what its fate is. So it's not going to stay in the blood very, very long. This is why I don't do it this way. It takes me forever. Secretions. And IgG stays in the plasma. So I'll just put a P here. It's in the plasma. This is in the plasma. So what about IgE? Now, most times you address the infection, it's gone before IgE gets secreted. So what I'm going to put for that over here is much later, and only if chronic inflammation is still going on. So if you're innate and these guys aren't able to address the problem, then you'll see IgE. So that's why IgE is correlated with conditions like arthritis, where you have chronic inflammation, or asthma, or allergies, because those don't tend to go away. You keep having this allergic response, or you have arthritis because you've worn down your cartilage, and you tend to see elevations in IgE if that's the case. So chronic inflammation tends to cause IgE to make more inflammation, and round you go. Okay, any questions on this one? On what I put, there's no lecture question that talks about this, but I do cover it in the lecture, so I do want you to know it. Although I kind of said for each type, give all the functions. Well, that doesn't really put them in what order are they released. Okay, any questions in chat? Okay. All right, let's talk about first the primary, secondary, and how each of these, let me see if I can erase just all this mess. Oops, I got all of it out. I guess I'll just erase all of it. I was going to try and leave that one box, but I got wild with my thing. So a primary response, and that's going to, we want to quantify this for the essay. So if you've never seen the pathogen before, that antigen hasn't been in your body, you've got to get your dendritic cell to get your T helper cell going that then can find the right B cell. That B cell has to have found the antigen, become an antigen presenting cell. All those events have to occur. And the book gives a range, and I'm not super crazy about exact numbers. And I don't know if I always say 7 to 10 days, but I don't remember what our book says before you see significant. And we're going to just talk about IgG because it's 70% of the antibodies in the plasma. So you're getting a good week. Some books will even say 2 weeks before you really get a peak. Are we good on that? So if you say 5 to 12, I'm not going to get crazy about it. It varies depending on the entry that the pathogen's coming in, where the B cell is. So there's a pretty broad range, but it's long. It's not a short time. When that primary response occurs, then from what I just said, which antibody is first released?
IgM.
IgM in low amounts. Okay, and we're going to call that low amount X. Okay, then IgA and IgG in relatively more amounts, low amounts. So if we're going to call that amount X, we're going to say maybe 10X here. So you're going to get maybe 10 times more IgG. I'm just making this up, right? Than IgM. But why I'm stressing this is because we want to compare it to a secondary this is for lecture question 20. Okay, so then we may or may not get IgE released, depending on, and it's only IgG that's more than these two. We'll call this one an X also. Are we okay? All right, so now you get say and it leads to IgG and not 10x, but 1,000 to 10,000. Did I get enough of them? X amounts. So not only do you get more, then you get about the same amount of IgM as you got here in the first one. Now you're getting, if that was 10x, this is 100 to 1,000. counts. This is what, they hope for, that you have some immunity after being exposed to COVID because you still have these IgGs for a couple months afterwards. And in a secondary, it's about 2 months. In a primary, let's put it up here. It lasts, IgG lasts a few weeks. I'll say 2 weeks, about 2 weeks. Now, it doesn't disappear suddenly, but it doesn't tend to be as long lasting. Now, you might say, well, yeah, but then they're saying maybe you have immunity longer than 2 weeks if you got COVID the first time. Well, you probably have more than one IgG type because the pathogen has more than one antigen. So, some are going to last and trigger later and last longer. But whether you have it for long periods is unclear. Now, a secondary, you would get a longer lasting, a more stable antibody. Okay, that brings us to our last topics, and that's active and passive immunity. We only have a few minutes for this. I do think the book does a good job with this. So, active immunity, and they never really define this, so I think this is a good way. Active immunity means you have memory cells. I don't care if they're B or they're T, you've got memory cells. Passive immunity means no memory cells. Instead, what you have, immunity via having antibodies. Well, how can you have antibodies without memory cells? Well, they're cells as antibodies. So the book divides up active immunity that you can get it 2 ways. So what are the 2 ways that you could get oops, that's not going to work, that you can get active immunity? Stupid thing. There we go. The one that you're not happy with is how do you generate these memory cells? And you're not happy.
I'm not happy with both of them, but one is naturally acquired and one is through vaccination. So naturally artificial.
Yeah, so naturally acquired means that you actually got sick,
yeah.
Right? You get sick.
I don't like vaccinations either, though.
Oh, well, you better start liking them naturally. That's the natural way of doing it. And then the other way is you get vaccinated. So that's where I spend a little bit of time, and I'm hoping in micro, they spend more time if you take micro, talking about what are conjugated vaccines, what does that mean, why do you need to do that, what are multi cocktails, like the flu one is a cocktail, a bunch of antigens, versus a subunit where you're getting one antigen, versus the killed or weakened whole organism ones. There are different advantages for these different kinds of vaccination. Okay, so not to go over, at least not much, there's also 2 ways that you can get passive immunity. And you should know that 2 different antibodies are involved. So one way is what? I think they call this one natural too. What does that mean? Anyone?
You get it through placenta exposure and lactation. I think placenta exposure is IgG and lactation is IgA with the colostrum.
Yes. So IgG crosses the placenta. And the baby gets long antibodies. or via IgA. Remember, milk is a body secretion through lactation. And in fact, you probably already know, or at least the women out there may know, that the first milk that's let down is no milk at all. There's no actual, mother's milk in that. When you nurse, the first milk that comes down is called the colostrum, and it's just a bunch of plasma extracted with lots and lots of different antibodies that you're giving to the baby. And that's one reason, natural breastfeeding is so beneficial to the baby. And that continues. It's not just that she stops secreting it. As long as you're lactating, you're secreting IgA antibodies into the milk. It's just not as concentrated as that first colostrum. So having someone not breastfeed at all, the baby is not getting those advantages. So a few people ask, why do people get sick with vaccines where others do not? Well, you know, when they're making the vaccine, you might get an innate response. And what you mean by sick is you might get a little bit of a fever. That doesn't mean that you're getting the disease. You're probably getting a reaction to could even be to what they're conjugating the antigen to. Conjugates are larger molecules that they stick the antigen on to try and trigger a stronger immune response. But that could be a stronger innate immune response. And you know, fever is part of that. That's part of your innate. So it could just be a reaction. Some people, since vaccines are often made in eggs, also are allergic to egg proteins, and they get the flu and the fever response as well, or an allergic response that can happen. Flu shots are only rolling the dice. So you can get the flu, but you're not getting the flu after a flu shot to the influenza viruses that are in that flu vaccination. You're getting some other flu. So you don't get the flu from a flu shot if you're being vaccinated. Don't be afraid of vaccines, people. We need them. Okay, how about the artificial way in which you can have passive immunity? Because we're going over here.
You get a gamma globulin shot.
Yeah, so you can tell gamma globulin shot. And I've met very few people who've gotten these. But if you travel to, or very few students that I've had, if you travel to areas where there's a lot of endemic diseases there that are serious, so sometimes they will give you, again, this big cocktail, kind of like the colostrum of the mom, and it's a big shot, a big needle and a big amount that they're putting in you. So you're not sitting on the plane too comfortably after you get this. But the idea is to give you a whole bunch of antibodies to a variety of some of these more serious diseases so that you'll have some chemical warfare to help you get through, and it won't prevent you from getting sick potentially, but it might make you get less sick than you would otherwise. It's only going to last a few weeks to months, so it's not any. As long as these guys hang around, it'll last. Okay, so lots of conversations about getting sick when they get flu shots. All right, so the last question is a whole bunch of scenarios. If you guys can work on those on your own, maybe when we do the review for the test, hopefully we'll have time to do at least some of the review, because now we've not only fallen behind, but we're way behind with this internet issue I had today. So if you can show up at 1 in just 5 minutes, run out to the bathroom, get something to eat or something, maybe we'll make it 1:05. What I thought I'd do there is introduce the respiratory slides, in particular the histology part of it. So if you can, that would let you work on the slides. if you cannot show up, you've got another class, or you've got to go to work, whatever reason, at 1:00 or 1:05, then work on the models, because the pictures in the lab manual are pretty good for all the respiratory models, and we can talk more about it. So I had scheduled a lab that is just studying for the review for the test, and I'm not going to get rid of that, but we might end up flip-flopping things around a bit. So to try and make sure that you get going in the lab, and don't fall behind, just skip doing respiratory. Try and work on that, because I may go much faster through the anatomy part of the respiratory in order to catch up our time. Okay, why don't you guys take about 10 minutes, and we'll see you if you can. We'll see you at 1:05. All right, I'm going to stop share so I can go to